Thank you, everybody. Today I'm going to present some work I did in UC Santa Barbara. I worked in a chemistry lab for a while, and this is about using conjugated oligoelectrolytes to increase power generation in E. coli and microbial field cells. So first part of my talk will be a brief introduction of what is conjugated oligoelectrolyte, their properties and synthesis. And then I'll talk about how to incorporate COEs into cell membranes. The biocompatibility of those COEs were also examined with the cytotoxicity test. And the final part will be incorporate COEs into cell membrane and put it in MFC, see how it improves MFC performance. So first, what is COE? COE is a synthetic molecule that's described by a pi conjugated backbone, and those two groups, the ionic pendant substituents at the end of the backbone. So in a pi conjugated backbone, the electrons in a pi orbital, which is perpendicular to other three sigma bonds, are highly mobile, making this part semiconducting. The two ionic pendant groups on the both ends of the backbone are ionic substituents that ions can easily associate and dissociate from the COEs. The structure of the COE makes it unique that it has a hydrophobic backbone with two hydrophilic ends. The structure is pretty similar to the conjugated polyelectrolyte, which has been studied a lot because their unique properties of semiconducting and water solubility. This molecules has been used a lot in optical electronic devices, such as organic solar cells, to use as a electron to reduce the electron injection barrier at the organic electrode interface, as well as reducing the operating voltages in some of the polymer-based light-emitting diodes. So you probably have noticed that the property of COEs is very similar to the cell membrane, which also have a hydrophobic backbone and hydrophilic ends. And the length of the bilayer is basically a shield between the inside and outside. It usually holds a thickness about, I would say, from 40 to 60 angstrom. It's pretty similar to the length of the COEs. And so this image shows that the COEs has been successfully integrated into yeast cell membranes as well as the lipid bilayer vesicles. Those are images taken with the excitation light source of a polarized light source. So if you take a detailed look at the images, you will see stronger and weaker emission profile. The stronger direction is in parallel with the direction of the excitation plane, and this weaker direction is perpendicular to the excitation plane. This confirms that the COE is aligned well in a membrane with the bilayer. So this method has been previously demonstrated to examine the local direction of liquid crystals. Here we design and synthesize two series COEs, COE1 series and COE2 series. In each series, we have three different chain lengths, which is described by how many aromatic rings in a backbone, named as DSBN, DSSN, and COE1-5C in a COE1 series, and in a COE2 series named as COE2, 3C, 4C, and 5C. This is by how many aromatic rings in a backbone. And the first one, the difference between the COE2 series and COE1 series is that COE1 series have those ionic terminated acyl chains stemming from tertiary amines, and this one is stemming from 4 alkoxy linkages. We'll put all 6 different molecules into the MFC and see how it improves the performance, and hopefully we'll find out how those structure variants will change or modify the COE performance. So I'll have a brief introduction of how to, this is a process of typical COE synthesis. And first you, sorry. Okay. First, we take 1 and react with the corresponding phosphonate 2 or 3, which generate the neutral precursor to COE3C and 4C named as COE23N and 4N. To synthesize COE25C, we first react 1 with equal molar of 2, which you have replaced only 1 terminate of this group, and then react with this chemical to generate the neutral precursor to COE25C. This is a typical synthesis, and it's a well-known process. And the COE1 series synthesis is pretty similar to this, and I'll not talk about the details here. So first, we are trying to see whether we can incorporate COEs into E. coli cell membranes. This is a video showing. So we prepare cells first, and we wash them several times in a buffer solution and put it on a polylysine-coated glass slide. And then we drop 5 micromolar DSSN into the solution. And then we take fluorescent images out of this immediately. And you will see that the staining process started immediately, and it became complete always in about 180 seconds. So it's a very fast process. And the right side image here showed a zoomed up image at 180 seconds. You can see that cell membranes are properly stained. Due to the small size of E. coli, it's hard to see the distinguished line. But we can confirm that the COE is aligned with the cell membrane with another method. The COE is when you dissolve COE in different polarized solution, it emits at different wavelengths. So if you dissolve COE in a water solution, which is a polar environment, it emits very weakly around 600 nanometer. But if you dissolve COE in a nonpolar environment, such as toluene, it emits strongest, very strong at 180 nanometer. And so when we observe the fluorescent the emission profile of COE stained E. coli, and we found out it emits very strongly at 480 nanometer, which confirmed that the COE is aligned well in a lipid bilayer, which is the nonpolar environment. We also confirmed we also did a Kirby-Bauer antibiotic testing, so to see how the COE will inhibit cell growth. So we prepared paper discs and soaked the paper disc with different type of COEs of different concentrations. So the concentration we tested is from 5 micromolar to 1 millimolar. And we prepared a cell plate, spread cells on it, and then attach the paper disc onto the plate, and then culture it overnight. And then we see whether an inhibition zone formed outside of the paper disc or not. So we found out that COE1 series didn't show any inhibition at a concentration as high as 1 millimolar, and same as COE23C. However, 324C showed a very minor inhibition zone at a concentration of 1 millimolar. And for COE25C, it showed a slightly larger inhibition zone at 100 micromolar. A cell cytotoxicity test was also done of COE with a concentration of 20 micromolar. We first prepare cells. We rinse it off with buffer solution, and then add COEs in the solution and dilute it with different dilution ratios. Then we culture it overnight and count the colony formed of each dilution ratio. And we picked up this dilution ratio and compared different COEs with the control without any COEs added into the solution. And we see that only COE24C and COE25C showed 30% off of colony formed on the plate. So This means that most of our COE we design are biocompatible, but only those two showed slightly toxic to the cells. But we'll see how those two behave in the later MFC performance. So we used the Microbial field cell array to screen all the COEs in MFCs. You probably have seen this platform in yesterday's poster. So this one consists of 5 layers. The first layer is the anode electrode layer, have 24 anode electrodes. And this is the anode chamber layer. This is the proton exchange membrane layer, the cathode chamber layer, and the cathode electrode layer. So the cathode chamber layer is a microfluidic cathode chamber layer, which has the cathode light inlet, which connecting all the 24 cathode chambers. We also have a control channel layer, which can close the valves while not replenishing the cathode light. And this is the device. It's about, the whole assembled device is about 3 by 3 inch. So this is a power curve when we add 20 micromolar COE into E. coli solution and add it into the MFC array system. We can see that all the COE-assisted microbial field cells demonstrated improved power generation, and among which the DSBN in COE1 series and COE25C and COE2 series show the highest performance, which is about 20-fold or 15-fold higher than the control without any COEs. So to roll out, there's the abiotic process brought in by the COE. We add COE only into the MFC system, and we test the power. We can see that there's negligible power generation compared with the one with microbe as catalyst. So it's true that the COE modify the cells and improve the power generation. So when we plot the polarization curve of this, we see that this is COE2 series. We see that if we calculate the slope of the linear region, we see the internal resistance has been decreased. About 20 to 30% the resistance. However, it's not very obvious for the COE1 series, and most of them were very close to the control or sometimes a little bit higher. So we can see that the COE structure makes some difference in the MFC performance, but it's just it's not clear for now. In another study, we tried different substrate. We used 40 millimolar lactate in this experiment. We can see that this is with control and COE25C, and the internal resistance has also been decreased. That might be the reason why COE2 series is improving the MFC performance. And remember, COE5C is slightly toxic to cells. We have also examined the biofilm formation on the electrode. This is, so because COE is fluorescent, you don't have to stain the cells in after you get the membrane, so it's autofluorescent. And you can examine the biofilm formation on the electrode directly. So we can see that COE1 series and COE2 series form different biofilm formation. And the morphologies are very different. So it's too early to talk about how or why the biofilm morphology changed for now, but it's for sure that the COE structure influenced the biofilm formation on the electrode. So I'll look to conclude what I'm talking about today. So we synthesize 6 molecules, and we test the cytotoxicity of the molecules. And because the cell fluorescent property of the molecules, we can examine the cell property and the biofilm formation directly without any staining process, and the power generation has been also improved. So our study provides the first fundamental picture of COE improving MFC performance. But following studies, such as the COE how they change the cell membrane, or how they change the cell to electrode interface, may also and it's also important to give a more mechanism picture of how COE function in an MFC system. I would like to thank Professor Guy Bazan for this very interesting project and all those lovely people working in the group. And also my PhD advisor, Dr. Han, provided me some initial materials for the study. And Dr. Brucey Logan found a generous support so I can stand here present this interest result, hopefully, to you. And also our funding agencies. I'm ready for your questions.
So I guess you're assuming the COEs are redox active. Is that correct?
Are what?
Are redox active?
Yeah. No, it's hard to say that. It might just create a tunnel in the membrane. So it's the mechanism of how COE is functioning in MFC is still under study. But it's definitely, if not be used as the molecule to improve the performance of MFC, such as neutral red or methylene blue, it might also be used as a tool to study the electron transfer pathways.
So if they were redox active, what would be the redox potential?
We haven't done that kind of study, but if I remember correctly, I'm not sure about that. I can check the detail for you. But we have done a CV test of these molecules on the graphite electrode, and it shows that the potential is around 0.3. But I'm not sure, so I can check the detail for you later.
So I have two small questions. Is there a specific reason why you use that trimethyl ammonium group at the terminal end? Because that's constantly positively charged. Have you ever tried a negatively charged COE?
Oh, I might. Let me see. So if you look at this one, so this molecule is actually neutral. So it has a positive charged nitrogen here, but it has a negative charged iodide here. So this is a neutral molecule.
But that ionizes normally, right?
Yeah.
So it would have more like a positive charge behavior or not?
Yeah, you're right. And we have also designed negatively charged molecules, but we haven't tested in MFC yet.
And a very small one, I may have missed this. Is it only inserting in the outer membrane, or may it have a tendency to migrate to the inner membrane as well?
I believe it's in both membranes. If you look at the, so it's hard to tell from this image, but sometimes the COEs can go into the membrane, into the inside of the cells. It's more clear in this east image. You can see some of the cells are staying inside, but we are still trying to understand why sometimes it's staying inside and sometimes only in the membrane. But for most of the cases, in the E. coli images I have taken, most of the cases only the membrane are staying.
I was curious if you ever watch the cells divide and see if it goes to the daughter cell as well?
We definitely see this in yeast cells. The COEs staying in the cells can be transferred to the daughter cell. And we are still not sure it's just directly transferred to the daughter cell. I believe it's that, but we cannot rule out another possibility that because we have COEs in the free solution, the new cells can uptake COEs from the solution too.